Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing University of Hyderabad MSc Entrance Examination question paper for the subject Molecular Microbiology conducted in the year 2020. Coming to the first question, identify the mismatch. So it is tuberculosis is a waterborne disease. Tuberculosis is an airborne disease. The answer is option D, that is the mismatch. Then second law of thermodynamics says no operator can operate in such a way that its only effect is to convert heat absorbed by a system completely into. Answer is work done by the system. Answer is option B. Then which of the following are some of the commonly used biochemical tests in microbiology? Identify the mismatch. So here it is phenol red acid production, phenol red malate broth. So that is the mismatch. So phenol red test is fermentation of carbohydrates, and carbohydrate source is phenol red malate broth. So the answer which is mismatch is option. Then identify the correct matches. Colonies produced by Pseudomonas on McConkey medium green colored. Fried egg appearance colonies on solid media by mycoplasmas. That is correct. Answer is option 2. And option 1, colonies produced by Pseudomonas on McConkey medium are pale colored. Then mycoplasmas reproduced by budding. It is not budding, it is by binary fission. And bacterial endotoxins are localized in lipopolysaccharides of gram-negative staining bacteria. That is also correct. So 1 and 2 and 4 are correct. Answer is option C. Then next one. The major reason that antiparallel beta-stranded protein structures are more stable than parallel beta-stranded structures are that the latter. Okay, so parallel beta-pleated sheets are type of beta sheets in which two polypeptide strands run in the same direction. And antiparallel beta sheets are type of beta sheets in which two polypeptide strands run in the opposite direction. So the major reason that antiparallel beta-stranded protein structures are more stable than parallel beta-stranded structures are that the latter have weaker hydrogen bonds laterally between adjacent strands. So the answer here is option D. Then next one. This type of questions, that is preparing of stock solutions and everything, that is a repeated question. Okay. They have repeated in so many molecular microbiology question papers. Then one scientist has asked his research scholar to prepare 100 ml of GTE buffer containing 1% glucose, 25 millimolar tris and 10 millimolar EDTA. He gave him glucose powder and stock solution of 1 molar tris and 0.5 molar EDTA. How much quantity or volume is required to prepare the required concentration of GTE buffer? So glucose, he has given the powder itself. So 1% is 1 gram in 100 ml. So for 100 ml you can take 1 gram glucose powder. Okay. So that is for preparing 100 ml of GTE buffer, you can take 1 gram of glucose. You can weigh and take. Then for the others, that is tris and EDTA, the formula is M1V1 is equal to M2V2. So M1 here is how much we have. That is the stock solution. What is it? So it's 1 molar, and out of that how much volume we have to take to get 25 millimolar tris or 25 into 10 raised to minus 3 molar tris for 100 ml. So we will get it as 2.5 ml tris. Similarly for EDTA you will get 2 ml of EDTA. So the answer here is option B, that is 1 gram glucose powder, 2.5 ml of tris and 2 ml of EDTA. Answer is option B. Then next one. A mutant cell loses parts of the end of its chromosome every time it replicates its DNA. You suspect that it has defect in it. So telomerase, that is an enzyme that works as a cellular reverse transcriptase that adds DNA in the chromosomal ends. It involves the protection of chromosome endings from getting shorter during replication of DNA to avoid the loss of catastrophic DNA. So the answer here is option D, that is telomerase. Then, which statement is incorrect about SDS page technique? Separation are run on gel covered plates. That is correct. Sodium dodecyl sulfate is used to denature the samples being separated. Yes. Proteins are denatured by sodium dodecyl sulfate or SDS. Then it is a type of gel exclusion chromatographic method of separation. Yes, it separate based on molecular weight. Then it is used to separate native proteins. Is SDS page is not using does not separate native proteins because you are denaturing the sample. That is, SDS binds to the protein and make them uniformly negatively charged. So when the current is applied, all SDS bound proteins in the sample will migrate through the gel towards the positively charged electrode. So it is used to separate native proteins. Answer is option D. That is incorrect. It does not separate native protein, it denatures the protein. So the native conformation is lost. The next one. Presence of DNA degrading enzymes in the extracellular medium would prevent transfer of DNA by natural transformation. So answer is natural transformation is the process by which bacteria can actively trap and integrate exogenous DNA, thereby providing a source for genetic diversity. That is natural transformation. So answer is option C. The next one. An example of oxidative deamination is, that is, glutamate equal alpha glutarate plus NH3. Answer is option C. So oxidative deamination is a form of deamination that generates alpha keto acids and other oxidized products from amine containing compounds and occurs primarily in the liver. So the answer is glutamate giving alpha ketoglutarate plus ammonia. Then dash is the operation one or more component. In dash operation, one or more components of a mixture of liquid or solid phases are transferred to another liquid phase. That is in case of extraction. Answer is option B. Then Raoult's law applied to ideal solutions. Answer is option. Then chaperones are concerned primarily with protein folding. Answer is D. Which of the following cells are epithelial cells located in the gastric gland? So they are parietal cells or chief cells. So answer is both B and D are correct. Then identify the correct match. Mesosomes are mesosomes are folded invaginations in the plasma membrane of bacteria. So they are not part of Golgi apparatus. Then enzyme hydrolyzing bacterial cell wall is not protease, it is lysozymes. And the photorespiration involves glycolate cycle. So that is correct. And bleaching is done by algae. So chemolithoautotrophic microorganisms only bleaching is done. So example is thiobacillus. So the answer here is option C, that is photorespiration involves glycolate cycle. So coming to the next question, identify the wrong statement. Albumin is not a hemoprotein. Then ciprofloxacin act as inhibiting bacterial DNA gyrase. Zinc is required for the enzyme alcohol dehydrogenase. Eye cell disease is associated with mitochondria. Eye cell disease is not associated with mitochondria, it is associated with lysosomes. So the incorrect or the wrong statement is option D, whereas all the others are correct. Answer is option D. The mode of nutrition of parasitic protozoa, it is holozoic. Answer is option C. Identify the wrong statement. So here, the genus Torula is a common yeast which is often grown on liquid in wood liquor and is a good source of amino acid glutamic acid. Then botulism is the most common foodborne disease caused by the spores of Clostridium botulinum, which produces the neurotoxin botulinum. Then rancidity of stored food is due to activity of proteolytic microorganism. It is not proteolytic microorganism, it is due to lipolytic microorganism. So that is the wrong answer. Answer is option C. Virulence of microorganisms can be reduced by attenuation. That's also correct. So answer here is option. Then identify the wrong statement. Vidal test is used for the detection of typhoid fever, which detects agglutinative creating antibodies against the O and H antigens of Salmonella typhi. Then Corynebacterium diphtheria is the pathogenic bacterium that causes diphtheria. It is also known as Klebsiella bacillus. Then acridine dyes are more effective against the obligate pathogens like mycoplasma. So it is not against mycoplasma, it is effective against gram-positive bacteria. So that is the wrong statement. Answer is option C. Then identify the wrong statement. Cefepime is an antibiotic which acts by inhibiting cell wall synthesis. Aminoglycosides are bacteriostatic. So aminoglycosides are not bacteriostatic, they are bactericidal antibiotics. So the wrong statement is option B. Then next one, which of the following statement is or are true regarding agrobacterium? Agrobacterium tumefaciens causes crown gall disease in monocotyledons. It is not monocotyledons, it is in dicotyledons. Then agrobacterium rhizogenes induces hairy root growth in dicotyledonous plants. That is correct. Then both agrobacterium tumefaciens and rhizogenes are gram-negative in nature. That is also correct. Both agrobacterium tumefaciens and rhizogenes use vertical gene transfer to infect plant. They use horizontal gene transfer, not vertical gene transfer. So 2 and 3 are only correct. So the answer is option C. Okay, next one. Which of the following correctly defines the primer? So the primers are short sequences at the end of nucleotide sequences which are used for amplification. Primers are short sequences which are complementary to the nucleotide at the ends of the sequence which is to be amplified. That is correct about primers. So answer is option B. Then match the following. Rhodobacter spheroides. So 1 is purple non-sulfur bacterium, 1,5. Then Chlamydomonas. Chlamydomonas is algae, 2,4. Heterotroph, 3, that is fungi, 2. And murine is bacterial cell wall, 4,1. And bacterial photosynthesis is hydrogen sulfide, 5,3. So the answer is option A, that is 1,4,2,4. Oh, sorry, 1,5,2,4,3,2,4,1,5,3. Then next one. Given below are statements on chromosomes and replisomes that are involved in prokaryotic DNA replication. Which of the above are correct? Okay. The chromosome is a helicase plus a DNA polymerase 3. Then replisome includes a chromosome plus 2 copies of DNA polymerase 3. That is correct. So 2 is correct. Then chromosome opens the DNA and creates RNA primers on lagging strand. That is also correct. Then a replisome coordinates replication on both the leading and lagging strands of the Y junction. That is also correct. So the answer is B, that is 2, 3 and 4. And chromosome is a helicase plus a DNA polymerase 3. Now chromosome consists of 7 proteins, that is DNA G primase, DNA B helicase, DNA C helicase assistant, DNA T, PRI A, PRI B and PRI C. So 1 is wrong, whereas 2, 3 and 4 are correct. So the answer is option B. Then next one. Consider the following statement that describe the function of the eukaryotic promoter. Which of the statements related to the above are correct? Okay. Typically lies towards the 5' region of the gene. That is correct. Then many eukaryotic genes have conserved promoter sequence called the Tata box. That is also correct. Answer is option. Two more are there. It is the region of translational start site. It is not translation, it is transcription. Then serves as sequence to which transcription apparatus bind. So answer is, the 4 is correct. So the answer is option B, that is statements 1, 2 and 4 are correct. Uricotelic animals are those that excrete nitrogenous waste in the form of uric acid. Which among the following is a uricotelic organism? So it is birds. Answer is option D. When DNA is denatured, it becomes single-stranded. Answer is option A. Then how many chiral carbon atoms are present in glucose molecule? So this is the glucose molecule structure. This is C1, C2, C3, C4, C5, C6. So the number of chiral carbon atom, that is, this is the second carbon atom to which OH, C3, C1 and H are attached. Then you have got the C3 carbon atom to which OH, C2, C4 and H atoms are attached. And then you have got the C4 one where H, OH, C3 and C5 are attached. And then you have got the C5 one to which OH, C4, C6 and H are attached. So in how many chiral carbon atoms are present in glucose molecule is, it is 4. So answer is option C, that is 4. Then next one. What are the amino acids which are biosynthesized from oxaloacetate? That is asparagine, methionine, threonine and lysine. Answer is option A. Then what is the function of sieve cells that are located in the root system? So they are located in the phloem and function as to transport organic compounds. Answer is option D. Then vas efferentia is a part of one of the most important human organs which arise from testes. Answer is option D. Then a DNA stretch of length 25.68 kilobase pair is equivalent to how many megabase pair? So 1 megabase pair is 1000 kilobase pair. So 25.68 kilobase pair will be how many megabase pair? So that will be 0.02568. Answer is option C. Then next one. Given that the molecular weight of NaCl is 58.44, how many grams of NaCl is required to prepare 0.3 molar solution? So here we have got the molarity given and the molecular weight is also given. For 1 liter, how much grams we have to take and dissolve. Okay, so that is WB. So this is molarity by WB by MB. WB is how much weight we have to take of the given compound. MB is the molecular weight of the given compound and volume in liters. So WB is equal to M into MB by V, or it is 17.532 grams. So the answer is 17.53. That is option C. Then next one. The process of translation requires the presence of, it requires mRNA, tRNA and ribosomes. Answer is option A. Consider the following statements comparing the organization and composition of prokaryotic, that is E. coli, and eukaryotic mouse ribosome. So E. coli has 70S ribosome made of 30S and 40S subunit. So E. coli has 70S ribosome consists of 50S large and 30S small subunits. Then next one, mouse has 80S ribosome made of 40S and 50S subunit. So it is a eukaryote. So eukaryotes have 80S ribosome consisting of 40S small subunit and large 60S subunit. So 1 and 2 are both wrong. Then 16S rRNA is present in the smaller subunit of E. coli ribosome. So that is correct. Then 18S rRNA is present in the smaller subunit of mouse ribosome. That is also correct. So which of the following statements are correct will be 3 and 4 are correct, or option C is the correct answer. Then the ratio of SDS to protein in SDS page, that is 1.4:1. Then effective chemotherapeutic agents are difficult to develop for the treatment of fungal infections because fungi have cell wall. Answer is option A. Then the purity of an enzyme at various stages of purification is best measured by specific activity of the enzyme. Answer is option. Then match the human syndrome given in panel A with chromosomal imbalance described in panel B. So Down syndrome, it is caused by an extra chromosome 21. 1C. Klinefelter syndrome, it is caused by an extra chromosome in males, and Part2 syndrome is caused by an extra chromosome of 13, and Edwards syndrome is caused by an extra chromosome of 18. So answer is option C, that is 1C, 2D, 3B, 4A. Then a special voice box which is characteristic of birds and is located on the posterior end of the trachea and its junction with the bronchi for producing sound is called syrinx. This is a repeated question. Then the sex of a child is dictated by the inheritance of a single Y chromosome from the father. Answer is option C. Then which of the following is not a dietary antioxidant? It is vitamin K. Answer is option B. Then lacteals are associated with the lymphatic system. Answer is option A. Then if a strand of a helix has 30% adenine, which of the following inferences is true? So it has got 30% adenine, so the opposite strand will have 30% thymine. Answer is option D. Because adenine base pairs with thymine. So 30% adenine means 30% adenine will base pair with 30% thymine in the opposite strand. Then next one. The synthesis of glucose from lactate, glycerol or amino acids is called. It is called as gluconeogenesis. Answer is option D. This is. Then next one. In mice, the yellow coat color is always found in heterozygous condition as homozygosity for the allele determining yellow coat color causes lethality. A cross is made between two yellow coat colored mice which has yielded 2/3 progeny that are yellow coat color and 1/3 progeny that are normal coat color. This is best example of. So here it will be naturally recessive lethal, because if it is dominant lethal, then the heterozygous offspring as well as the homozygous offspring with the dominant gene will die out. So you will not get this particular ratio, that is 1/3 and 2/3. So it is recessive lethal allele. So this is the heterozygous condition, and both are crossed. These are the gametes. These are the offsprings of which small y will die out. So the answer here is option A, recessive lethal allele. Then next one, match the following. Ramachandran plot, that is torsional angles 1, 3. Protein sequence, Edman degradation 2, 4. Dialysis, purification 3, 1. Enzyme 4, substrate 4, 5. Sorry. Ribose 5 phosphate, pentose phosphate pathway. So answer is option C, that is 1, 3, 2, 4, 3, 1, 4, 5, 5. Then cyclostomata is a class of vertebrae, a class in vertebra. Answer is option A. Then antiviral drug vidarabine is. It is a nucleoside analog, inhibits synthesis of viral DNA. So answer is option A. Then match the chemical mutagen with their respective mode of action. So 5-bromouracil. It is thymine and base analog. Proflavin is intercalates between base pairs. B5. 2-aminopurine. C is 1 adenine base analog. D ethyl ethane sulfonate. D2 removes purine rings. And E nitrous acid. It converts C to U and A to hypoxanthine. So answer is option A, that is A3, B5, C1, D2, E4. Then A and B genes are very tightly linked and no crossing over occur between these two loci. Drosophila fly of the genotype capital A small b small a capital B is test cross to small a small b. What proportion of the progeny will be small a capital B small b? So these are the genotypes which are crossed. So these two genes, they are linked together and no crossing over take place. So only two type of gametes will be produced by this particular offspring, that is capital A, small a, capital B. And this particular parent will produce only one type of gamete, that is small a, small b. And upon crossing you will get 50% capital A, capital B, small b, and 50% small a, and capital B, small b. So our question here is for small a, capital B, small b, that is 50%. So answer is option B, 50%. Then immunoglobulin that results in histamine releases, that is IgE. Then identify the target cell receptor for cholera toxin. It is ganglioside. Answer is option D. Then higher plants are unable to fix dinitrogen because they do not have genes which encode dinitrogenase enzyme complex. Answer is option D. Then when boundary of a system does not permit the transport of matter between system and surrounding, then the system is said to be closed system. Answer is option B. Then Barbara McClintock first discovered transposable elements by experimenting with siamase. Answer is option B. The enzyme fumarate hydratase is a lyase. Answer is option B. The mechanism by which resistance to a virulent strain of a plant pathogenic virus is conferred by prior exposure of plant to a less or inactive form of the virus is known as. It is called cross protection. Answer is option C. Next one. Which of the following statement is true about amino acid? All protein forming amino acids are chiral except glycine. That is true about amino acid. So answer is option A. Chlorofluorocarbon pose a danger to the natural concentration of ozone. Answer is option D. Some stereoisomers are mirror images of each other. They are called enantiomers. Answer is option B. Usually the two sugar molecules bind with glycosidic bond. What type of bonds join sugar molecules? So it is covalent. Answer is option D. Then next one. If the average molecular weight of one amino acid is 110 Dalton, the molecular weight of a peptide made of 10 amino acids is expressed to be. So one amino acid it is 110 Dalton. So 10 amino acids will be 1100 Daltons. But one molecule of water is removed when two amino acids form a peptide bond. And molecular weight of water is 18. So when 10 amino acids form a peptide bond, 9 molecules of water are released. So the molecular weight reduces by. So 18 into 9, that is equal to 162. So 1100 minus 162, that will be 938. So the answer here is option B, that is 938. Then next one. The peptide alanine, arginine, glutamine, methionine, threonine, tryptophan, lysine, valine was digested with cyanogen bromide to produce. So cyanogen bromide, it cleaves at the C terminus of methionine residue. So it will cleave over here, okay, this region, and you will get alanine, arginine, glutamine, methionine plus threonine, tryptophan, lysine and valine. So the answer is option A. Then consider the statements about sex determination in Drosophila and choose the correct answer. So the fly with the sex chromosome complement X0 and 2 set of autosomes are sterile males. So one is correct. The fly with the sex chromosome complement XXY and 2 set of autosomes are fertile males. They are normal females. So 2 is not correct. The fly with sex chromosome complement XY and 2 set of autosomes are fertile males. 3 is also correct. And the fly with sex chromosome complement XX and 3 set of autosomes are intersexes. 4 is also correct. So the answer is option B, that is statements 1, 3 and 4 are correct. Then next one. The solution has a pH of 3.5. What is its pOH? So pH plus pOH is equal to 14. They have given pH of 3.5 and pOH we have to calculate. So 14 minus 3.5, that is 10.5. So the answer is option D, that is 10.5. Then next one. From the animal taxonomic study, some of the examples or characteristics are listed in column A. LMNO. Match its best combination with corresponding phylum present in column B 1, 2, 3, 4, 5 and select the correct answer. So, Cycon L4. That is Porifera, M1 Aurelia, that is Nigeria, and Bioluminescence. It is Ctenophora N3 and Octopus O5, that is a mollusk. So the answer is option A, L4, M1, N3, O5. The next one, a decrease in 2,4-D will cause an elongation of roots. Answer is option C. The insecticide Gamexine it is benzene hexachloride. Answer is option C. Then association between the aphid and the gamma proteobacterium is best example of. It is an example of mutualism. Answer is option A. Then one of the following statement is followed by the saturated fatty acid. That is, they have high melting points. Answer is option A. Then what is the primary function of gallbladder in human digestive system? So it stores and concentrates bites. Answer is option D. Then G.W. Beadle and Tatum were awarded Nobel Prize for Physiology and Medicine in the year 1958 for demonstrating that all biochemical reactions were controlled by genes and that each gene was responsible for the synthesis of a specific enzyme. This Nobel Prize winning research work was based on the experiment carried out with. So they have carried out the work with oxotrophic mutants of Neurospora. Then, in proteins, end-linked oligosaccharides are attached to asparagine. Answer is option D. This is a repeat one. The following bacterium is well known for production of the enzyme coagulase, which coagulates the fibrinogen in plasma. The clot protects the pathogen from phagocytosis and isolates it from other host defenses. So the answer is Staphylococcus aureus. Answer is option A. Then the IUPAC name of methyl cyanide is ethane nitride. Answer is option D. The stage of parasitic protozoa that actively feed and multiply are called. They are called trophozoites. Answer is option C. In an antibody Fab fragment, it binds antigen. Answer is option C. The next one. Each cycle of beta oxidation produces one FADH2, one NADH, and one acetyl-CoA. So the answer is option B. A man with heterozygous blood group A has children with a woman who has blood type AB. So man is heterozygous blood group for A. So its genotype will be AO and the woman they have told it is AB. Their children cannot be. So these are the gametes produced by the man and these are by the woman and you get these as offspring. So you will have the A blood group with genotype AA and AO, which is also of the genotype phenotype A, and AB will be AB blood group and BO will be B blood group. So the man cannot carry blood group O. So the answer here is option B, that is blood group O. Then next, which among the following is a non-proteinogenic toxic amino acid, that is cannavanine. This is also a repeated question. Then when benzene reacts with ozone to give triozonide as an intermediate, which on treatment with ZnH2O yield glyoxal. So answer is option B. Then when a leaf is dropped into a bucket of water, it floats on top of the water. Which of the following properties of water could explain what you observe? So it has strong surface tension. Answer is option A. So a cross is carried out between genotypes. They have given the genotypes. How many genotypes of the progeny are possible? So when you see the A locus, you have got capital A small a crossed with capital A small a. These are the offsprings which for the A locus. They can have this, how many genotypes? 1, 2 and 3 genotypes are possible. And how many phenotypes? Capital A and small a phenotypes. So 2 phenotypes are possible. Similarly, for the B locus, you have got one phenotype and two genotype. For C locus also two genotypes and two phenotypes. For D locus also one genotype and one phenotype, and E locus also three genotypes and two phenotypes. Now they have asked only for genotypes are possible. So the number of different genotypes possible will be 3 into 2 into 2 into 1 into 3. So that will be 36. So the 36 possible genotypes are possible for this particular cross. So answer is option A, that is 30. Then which of the following statements regarding structural gene? Consider the following statements regarding structural genes. Which of the statements given above are correct? So they encode protein that play a structural role in the cell. So that is correct. Then next one. They encode protein that interact with other DNA sequences. That is not correct. So a structural gene is a gene that codes for any RNA or protein product other than the regulatory factor. That is a regulatory protein. Also encoded by structural gene are non-coding RNA such as rRNA and tRNA, but excluding any regulatory miRNAs and siRNAs. So they do not encode protein that interact with other DNA sequences. So the next one, they contain sequences of DNA corresponding to the amino acids of a protein. So that is also correct. And they encode rRNAs. That is also correct. So 1, 3 and 4 are correct. So the answer here is option B. Then which of the following groups of hormones are produced by pituitary gland posterior lobe? So they are vasopressin and oxytocin. Answer is option D. Then which of the following will typically have higher 260 by 280 ratio in a microvolume UV visible spectrophotometer if the length of the fragment in each case is same? So a 260 by 2 ratio of 2 indicates a pure RNA, and if it is 1.8 it will indicate a pure DNA. The actual ratio will depend on the composition of the nucleic acid, that is RNA will typically have higher 260 by 280 ratio due to higher ratio of uracil compared to that of thymine. So the answer here is the complementary RNA obtained from AT rich single stranded DNA given in option A. So they have asked here which of the following will typically have higher 260 by 280 ratio. Usually RNA has got higher 260 by 280 ratio. Why? Because it has higher ratio of uracil compared to that of thymine in DNA. So when you have an RNA which is complementary to this AT rich single stranded DNA, that means RNA, instead of the T it will be U. So the uracil will be in higher ratio, and so this complementary RNA obtained from AT rich single stranded DNA will have higher 260 by 280 ratio. Then next one, which of the following forms of DNA will migrate faster in agarose gel electrophoresis? The condition in the molecular weight of these forms of DNA is equal. So they have asked which of will migrate faster in agarose gel electrophoresis. So it is supercoiled circular DNA that will migrate faster in agarose gel electrophoresis. It has less effective volume than others, thus it migrates more rapidly when subjected to electrophoresis due to its compact structure. So the answer here is option D, that is supercoiled circular DNA. Then kinase reactions. So they involve transfer of a phosphate group. Answer is option B. Then next one. In a biochemical reaction, an oxidizing agent gets reduced by undergoing one of the following changes of state. That is gaining electrons. Answer is option B. Then human immunodeficiency virus, HIV, that causes acquired immune deficiency syndrome, that is AIDS. It is a retrovirus. Answer is option D. Next one. Which among the following is a polymer of hydroxy fatty acids? So it is Q10. Answer is option B. Then the fovea of the eye, it provides the highest and clearest vision. So answer is option A. Then choose the correct answer from the given statements. So Azolla is a small aquatic moss that harbors Anabaena in pockets within its leaves. Azolla is not an aquatic moss, it is a fern. And Azolla will have filaments of Anabaena living within ovoid cavities inside the leaves. So the statement 1 is not correct. Azolla has proven useful as green manure in rice paddy fields. That is correct. So statement 2 is correct, whereas statement 1 is false. So answer is option. Then which of the following helps in getting a three-dimensional picture of the specimen? So it is the scanning electron microscope. Answer is option C. Recently, who has declared the COVID-19 as global pandemic? Which of the following information is correct regarding COVID-19? So it is genetic material is positive sense single-stranded RNA. That is correct. So answer is option A. The next one. The protein energy malnutrition observed in infants occurs due to simultaneous deficiencies of proteins as well as low energy levels or calories and is known as marasmus. Answer is option C. This is a repeated question. Then next one. If a DNA fragment is digested by restriction endonuclease in 4 sites, giving rise to fragments of which 3 are of equal length, how many bands would be seen after electrophoresis? So this is the DNA fragment and it is digested with restriction endonuclease in 4 sites. That is 1, 2, 3 and 4. How many fragments you will get? You will get 1, 2, 3, 4 and 5. A, B, C, D and E. So they have told that out of these 3 are of equal length. So A, B, C are of equal length. So they will separate out together. Suppose it is 2 kb fragment, all ABC will be 2 kb and you will see a comparatively thicker band near the 2 kb region. So these 3 will separate out together. Then you have got D and E fragment. That is D over here and E over here or anywhere. I have just drawn it just for understanding purpose. Okay. It can be either greater than this molecular weight of the D can be either greater than this particular band or even less than this particular band or anything. You will get 1, 2 and 3 bands. 1, 2 and 3 bands because A, B, C they are all bands of the equal length. So the answer here is option B, that is 3. Then next one. Identify the wrong statement corresponding to Robert Huerman-Koch findings. So he has established the causative relationship between a microorganism and disease. Microorganism can be isolated and grown in pure culture from the diseased organism. When introduced into a healthy organism should cause the disease, and the same microorganism must be re-isolated from the experimental host, which must be identical to the original specific causative organism. So A, B, C are correct. Then his discoveries helped in identifying the bacteria that cause anthrax, cholera, tuberculosis and salmonellosis. So he was a German bacteriologist who discovered the bacteria that cause anthrax, tuberculosis and septicemia and cholera. So the answer here is option D, that is incorrect. So option D is the answer. Then next one. With respect to the surrounding membrane system, which is the odd one out? So vacuole, chloroplast, endoplasmic reticulum, peroxisome. So chloroplast is the odd one out. Why? Because all the others are single membrane, but chloroplast is double membrane. So answer is option B. So that's it. Thank you for watching my channel. Please like, share and subscribe. Thank you.